I'd like to introduce our first speaker, Associate Professor Prue Francis, who is providing an update on the Breast Cancer Trials Research Program. Prue is the Head of Medical Oncology in the Breast Service at the Peter MacCallum Cancer Centre and a Consultant Medical Oncologist at St Vincent's Hospital in Melbourne. Prue is also the Chair of the Breast Cancer Trials Scientific Advisory Committee, and she joins us from Melbourne today. So hi, Prue.
Hello, Anna. Thank you. Thank you very much. And it's my pleasure to be here today. And as Chair of the Scientific Advisory Committee, I just wanted to give you a little bit of an update and some insights into some of the research that we're involved in at the moment and how we operate as a committee. So our committee has 26 members, and our members are actually from all the various states in Australia and also members from New Zealand. And we have a range of expertise in our Scientific Advisory Committee, so we're fortunate to have input from breast surgeons, from medical oncologists, radiation oncologists. We also have several translational researchers, and for those who are not familiar with that term, translational researchers are oncologists who not only see patients but also run a research laboratory in which they're studying certain aspects of cancer to try and give us new insights into clinical trials that we might conduct in the future. We also importantly have consumer advisory panel members who will provide us with a consumer perspective on the things that we're considering studying. And importantly, we have also inputs from a psychologist, from statistician, pathology, and a chief operating officer. And we also we have hundreds of other members that we can call on when we want to have reviews of various aspects of our trials that we're considering, and also our whole consumer advisory panel can be very helpful when we're thinking about trials for the future. So what is our mission and our vision? So for the Scientific Advisory Committee, we're aiming to conduct the highest quality clinical trials research so that we can produce better treatment outcomes for people who are affected by breast cancer. And our vision is that we can improve and save the lives of people affected by breast cancer, and also to try to prevent breast cancer from occurring. Now, for those who are not familiar with the nuances of breast cancer, increasingly oncologists think of breast cancer as being of different subtypes. So when I first trained in oncology, we more or less viewed breast cancer as being one sort of type of cancer, but nowadays we know that there are different subtypes. And I've listed some of the subtypes that oncologists may talk about that we use as a sort of a shorthand. So we might talk about luminal breast cancer, and that refers to a cancer that is hormone receptor positive or estrogen receptor positive. And then within luminal breast cancers, there are again a couple of different subtypes that oncologists will think about. And we refer to those as luminal A breast cancers. And those tend to be the hormone sensitive breast cancers that tend to grow more slowly. And those are ones where we feel that hormone treatment is particularly important as far as trying to control that cancer, after surgery. And then there are luminal B cancers, which are also estrogen receptor positive, but those are cancers that may have a somewhat faster growth, even though they're estrogen receptor positive. And these luminal types of cancers are the ones that we might consider somewhat less aggressive in their biology, particularly the luminal A cancers. And then some of the other subtypes have traditionally been considered more aggressive in that they can sort of grow faster and they can seemingly appear in the breast and sometimes be quite large when the woman wasn't aware of any change at all, and then one day there seems to be suddenly a larger lump there. So HER2 positive cancers is another important subtype, and then triple negative breast cancer. I'm going to talk a bit more about a trial in triple negative breast cancer. But for those of you who are not familiar with this terminology, triple negative breast cancer refers to a breast cancer that when the pathologist has tested the tumour sample, they found that it doesn't show any staining for the estrogen receptor in the cancer cells. There's no staining for progesterone receptor, so we call that negative, meaning there's no staining. and there's also no overexpression of HER2. So triple negative really means an absence of those markers in the pathology lab. Now, the other thing that's quite interesting nowadays in oncology is the immune system. And in breast cancer, we've been thinking more about how the immune system might be involved in breast cancer. And the current thinking is that the immune system may be more important in the control of certain types of breast cancer. So you remember we talked about the hormone receptor positive or estrogen receptor positive luminal breast cancers before, and those ones particularly with the slower growth. And we think that in those cancers, perhaps the immune system is a bit less relevant in the short term, whereas in some of those more aggressive types of breast cancer or subtypes, the HER2 positive or the triple negative, our current thinking is that the immune system is actually perhaps far more relevant in terms of how the body might control those types of cancer. So looking at triple negative breast cancer in particular, so that's the breast cancer that doesn't have any estrogen receptor or progesterone receptor, so no hormone receptors and no overexpression of HER2. If the pathologist looks under their microscope at a sample of the breast tumour, either in a biopsy or from surgery, and I've shown you in this panel on the right here some pictures of what that pathologist might see under their microscope. And the pathologist, with using special stains, can actually estimate the percentage of lymphocytes. And lymphocytes are a type of immune cell, and they can look and count, what the percentage of these immune cells is in the tumour. And basically in triple negative breast cancer, it's actually been found, and in particular by one of our researchers, Professor Shireen Loy, that if you have more lymphocytes in the tumour, and they're referred to in shorthand as TILs, or tumour infiltrating lymphocytes, in triple negative breast cancer, that's associated with a better outcome or a better prognosis. And so somebody who has 90% of these tumour infiltrating lymphocytes is likely to have a better long-term outcome than perhaps someone with 1% of tumour infiltrating lymphocytes. So that's not to say that somebody with a low number of the tumour infiltrating lymphocytes can't be cured, but if we were to estimate on average, it's better to have more of these lymphocytes or these immune cells in your triple negative cancer sample. So in thinking about these cancers, how can we tackle them apart from surgery and radiation? From a drug perspective, well, we know that triple negative breast cancers don't benefit from hormone therapy because they don't really have these hormone receptors, and they don't really currently benefit in the mainstream from any targeted therapy. For example, HER2 positive cancers can benefit from HER2 targeted therapy, but we don't have widespread targeted therapy available for these triple negative breast cancers. So in fact, for women or patients who have a triple negative early breast cancer, our main drug treatment really is chemotherapy. And generally that will go for at least 16 to 20 weeks. So it can be quite a long period of time that chemotherapy is prescribed in the postoperative or the preoperative setting for a triple negative breast cancer. But one of the things we've been wondering about in our group is whether we can use the patient's immune system to be smarter in how we treat these triple negative breast cancers. So I want to talk to you about a trial that we very recently launched, and it's called the NEON trial, and it's specific for patients who are diagnosed with a triple negative early breast cancer. Now in this trial, we're looking at patients having chemotherapy prior to their surgery, and we often do this in triple negative breast cancer nowadays, but instead of going for 16 or 20 weeks of chemotherapy, we've opted for a shorter block of chemotherapy, which is 12 weeks. And all the patients in addition will receive some immunotherapy treatment with the goal of trying to get the immune system a bit more active in the patient and perhaps get a better outcome from combining the chemotherapy with revving up the immune system. And in this trial, we're looking at a couple of different strategies for the timing of the immunotherapy And in one group of patients, we're going to give a single dose of the immunotherapy before we start the chemotherapy and some further immunotherapy. And down below, you can see another group of patients who'll start both those treatments at the same time. And all the patients in this study will get 5 doses of the immunotherapy, but the timing will vary a bit according to which group they're in. After they've had this period of treatment, then all the patients then would be next having their surgery, and then we'll be looking at what cancer, if any, is left behind, and then further chemotherapy or other drug therapy, radiation would be optional depending upon what was appropriate for that patient, and all the patients will be followed up for 3 years. So the primary endpoint of this trial is trying to see the percentage of patients who have no cancer remaining after their drug treatment before surgery. So if we look at this NEON trial, if you had a patient who had no evidence of breast cancer remaining in their breast tissue that was removed at surgery, it's possible that some of those patients could avoid some of the usually longer chemotherapy that would be given. And we also plan to study in these patients from their biopsy samples if the percentage of the tumour immune cells that we see in their biopsies, both before the drug treatment, during the drug treatment and after the preoperative drug treatment, whether that's clearly related to the outcome. So I thank you for your attention. I hope that that's given you a little bit of an insight into some currently very exciting research that we're doing in triple-negative breast cancer, trying to harness each patient's personal immune system so that it can give them the best chance to remain free from their cancer in the future. So thank you for your attention. I'll be happy to take questions.
Thank you, Prue. That was fantastic. I feel like I know a lot more about lymphocytes than ever before. We've had a question that's come through, Prue, from Diane, who has asked about treatments available for metastatic patients with HER2-positive breast cancer.
Right, so in fact, if you recall in the slides, I showed that HER2-positive breast cancer is also one of the cancers where we think the immune system may be important. And we've actually involved in 2 trials in metastatic breast cancer. One is currently active at the moment, and that trial is called the DIAMOND trial. And in that trial, patients with metastatic HER2-positive breast cancer who've received prior standard treatments and their cancer still has become active again. In that trial, patients are receiving a combination of 2 immunotherapy treatments as well as the standard HER2-targeted therapy with trastuzumab. And in this trial, we hope that by combining 2 different types of immunotherapy with the standard HER2 therapy, that we might be able to find a chemotherapy-free combination that might allow a further period of cancer control for that patient with metastatic HER2-positive breast cancer.
Well, that sounds like really interesting research.
Yeah, and the other interesting initiative we have that is coming, we hope starting this year for patients with HER2-positive metastatic breast cancer, is a trial called Together. And that will be looking at engaging a new oral drug called tucatinib in combination with immunotherapy and a HER2 therapy. So we're very excited about that and looking forward to starting that trial.